All right, everyone. I think we should now be on our YouTube channel as well. So it's a great pleasure for me to have my dearest friend, a brother in arms, Professor Seamus Martin. Seamus is one of the most highly cited scientists in the world in the field of cell death. He's also the holder of the Smurfit Chair of Medical Genetics within the Smurfit Institute of Genetics at the Trinity College Dublin. I had the wonderful pleasure to share the lab of Doug Green at the same time doing postdoc along with Seamus. From whom I learned a lot of things. Seamus has many prizes and honors. I'll be very short, just mention a couple of them. So he was elected to the Royal Irish Academy in 2006 and to the European Molecular Biology Organization, the EMBO, in 2009. And received the RDS Irish Times Boyle Medal in 2015, which is the Ireland's highest scientific honor. So Seamus is well known here in Brazil as well. He came to participate in this particular course from the beginning. I think is 1999 or 2000, something like that. And it's always a pleasure, although we are far away, at a distance. It's always great to see you, Shay. So we miss you a lot in Brazil. Hopefully you will be able to come very soon. Thank you so much. And I'll leave the audience to you. I want to thank all the presence also of the students here at the Zoom, as well as some of our speakers and colleagues also will be joining the Zoom room. But all the lectures, not all the lectures, many of our lectures will be also broadcasted to the YouTube. So yeah, thank you very much.
Okay, I'm starting now, am I, Gustavo? Just checking. Yeah, it's over to me. Can everybody hear me okay? Maybe if...
Yes, everything is cool.
Okay, that's great. All right, so I'm going to talk about programmed cell death, and I'm going to talk about really some of the reasons why we have programmed cell death. And, a lot of that is tied up in the effect cell death can have on neighboring cells and also on our immune system. And then I'm going to talk about basically some of the history of, what we've discovered and how we discovered it in different model systems, and to take us up to the present day in terms of how cell death control works. And I'll also maybe point out as I go along some kind of gaps in the knowledge and where things are going. So I've quite a bit of material. So at some point, if anybody wants to ask a question, I'm very happy to, respond if you want to, just call out during the discussion, or I can wait till the end. All right, so I work in Trinity College Dublin, as Gustavo mentioned. Sorry, I should say, first of all, just to thank Gustavo for the very nice introduction, and Karina, and also Ricardo for organizing the course. And yes, I think I've been giving a course in cell death at USP since, it probably is 1999. And of course, it's always wonderful to visit Sao Paulo and Brazil. And I very much miss not having been able to go there in the last few years, but certainly hope to in the near future. So this is Trinity College in Dublin, and we're just in the center of Dublin, which is very much on the coast of Ireland, on the east coast of Ireland. And it doesn't rain quite as much as people say it does, but it is a lot cloudier than Brazil, and it's quite green. And the Trinity College campus is about 400 years old, and it's quite a nice place to work. This is another view of it from inside the campus. And that's my office right there, just above the entrance arch. No, it isn't. We're at the back of the campus with the science buildings. So as you all know, the cell is the basic unit of life. And, I'll put this up just to remind ourselves that although we think of cells as quite simple things, of course, in reality, cells are really highly complex. And I think we're still 20 to 30 years from really understanding how it is cells do what they do. And what I mean by that is from understanding exactly how, basic processes like cell death works and cell division, differentiation, and many other processes that are fundamental to cellular life. An average cell has about 20,000 different proteins, and of course, hundreds of thousands of copies of each of those proteins, all working in different molecular complexes, with many proteins having many different other proteins that they interact with at different times, during the day or during whatever process they're working at. So cells are complicated, and also, of course, interactions between cells are complicated. And in order to fully appreciate why we have regulated cell death, we have to understand not just events that take place within the cell, but also events that take place between cells, which I'm going to outline in a moment, in quite anthropomorphic or human terms, in terms of how we behave as individuals within our human society. In many respects, many of the same rules apply to cells within their cellular society. So cells live in societies, in multicellular organisms, and in the human body, we have 10 trillion cells, which is a lot of cells, obviously. Now, we can simplify this and just imagine the body as a box, with the outside of the box, of course, being our skin. And then there's a hole running through the box, which is our gastrointestinal tract, which is effectively outside the body also. And then we have our respiratory system, our lungs. And then within the tissues in the body, we have a lot of cells. And we just think about this in very basic terms. Some things that we either intuitively know or we've learned over the last 50 to 100 years of studying cells is that, of course, there are some basic rules that cells need to obey to stay within the cellular society. So first and foremost, cells need to do their job. If they don't do their job, as we're going to find out, typically what happens is that they're killed, they're weeded out. And we'll get to exactly what the mechanism of that is, or how exactly they're killed. But fundamentally, we understand that cells need to do their jobs. And if they don't, they're not tolerated for very long. Another fundamental rule is that cells stay where they're born. That's a rule. A cell in the pancreas doesn't decide to have a vacation in the kidney. And a cell in the skin doesn't, again, go to visit the brain and then come back and, resume its normal life. Now, the exception to that is, of course, cells have an immune system that patrol the body looking for infection and looking for errant cells or cells that are behaving abnormally. Those cells do get to basically go to other locations in the body. But in general, the vast majority of cells need to stay where they live. They have molecular address codes in the form of integrins that contact the extracellular matrix uniquely in their area. And if they move, if they detach and they start to try and travel elsewhere, then typically they die. So, cells have to stay where they're born. Another fundamental rule, of course, which we, again, intuitively understand, is that if cells are living together, then they don't eat each other. And so a very basic rule is that you don't eat your neighbor in a cellular society. Now, as I'm going to tell you, that rule actually breaks down when cells die in a program manner. They do actually eat each other. And I'll tell you what some of the triggers for that are. But during programmed cell death, cells give out signals that effectively represent permission for their neighbors to eat them. But under normal circumstances, of course, cells don't eat the neighbors. The other thing is that they're not allowed to, let's say, build a new house. If in human society, you have a house, you can't just build another house, even if you have room for it on your property. You need planning permission, you need permission from the local authority to build. And in the same way, cells don't just divide spontaneously, they need permission to do that in the form of growth factors. So, that's another fundamental rule. Otherwise, we would get cancers all the time. cells are not allowed to harbor infectious agents, to harbor bugs. If they do, they're killed. And finally, and this is, obviously a very general rule, don't act weird. And what I mean by that is, in molecular terms, if a cell is expressing or displaying any peptides that the immune system doesn't like, that the immune system recognizes as non-self, the cell is deemed to be effectively acting weird and is also eliminated. So in the cellular society, it's kind of like human society. There are rules to live by. And in the cellular society, typically, if cells transgress the rule, then they're killed. And there are lots and lots of situations where cells basically die as a result of transgressing these rules. So if you don't perform your job in a cellular society, typically death is the penalty. And as I'm going to explain to you, the most common form of death that we see is programmed cell death, or apoptosis. Now, I'm going to touch on other types of cell death, but in the main, I'm going to talk about programmed cell death as being apoptosis, rather than, there is a programmed form of necrosis that has been recognized in recent years, but really it doesn't apply in the situations that I'm talking about in most of these cases. We're talking about apoptosis. So in a cellular study, if you don't perform your job, if you're injured or infected or abnormal or dysfunctional or breaking the rules in some way, typically we see that death of the cell is the penalty that's invoked. And the programmed cell death, as I said, that we're going to be looking at is apoptosis. And this is actually just to start off at the beginning, showing you what apoptosis looks like. The two cells in green here with large blue nuclei, cells have been stayed with DAPI, and also their mitochondria are stained here in green, the mitochondrial network. So these two cells on the bottom on the left are both healthy normal cells. And then these cells here, the cells that, have multiple fragmented pieces of nuclear material, that's very characteristic of apoptosis, or an apoptosis or programmed cell death. Cells condense their nuclei, the nucleus breaks up into many pieces, the cells undergo blebbing, but these cells are not actually technically dead. They're not leaking out the contents, they haven't burst open, and they're actually in the relatively early phases of programmed cell death. And in fact, in the body, normally these cells would already have been eaten by their neighbors, or eaten by resident macrophages, or macrophages that have come into the tissue. And in many respects, that's exactly what apoptosis facilitates, is the early recognition or removal of a cell that needs to be removed for one of the reasons I've already mentioned. So programmed cell death enables cells to be removed from tissues that have broken one of the rules of cellular society. And critically, it allows cells to be removed before they can burst open and release their contents, for reasons that I'll go into in a moment. So our tissues are undergoing this kind of constant throughput of cells being born and then cells dying for one of the reasons I've already mentioned, injury or aging or dysfunction or getting infected. And you have then cells typically undergoing programmed cell death in one of these various situations, for reasons we'll discuss. And that's very important, that most of the time the death is programmed, it's regulated, it's controlled, because there is another form of cell death that until around maybe 40 years ago was thought to be the main form of cell death. And that type of cell death is unprogrammed, typically it's called necrosis. And the endpoints between apoptosis or programmed cell death and necrosis are really very different. So there's this huge throughput going on all the time in all of our tissues, that cells are being born, they do their job, and then obviously over time they get injured or infected, age, dysfunctional, and then they undergo programmed cell death. And it's thought in some tissues that there is 1,000-fold turnover over our lifetime. In some tissues, of course, cells have very short half-life. Human peripheral blood neutrophils have a half-life of between 24 and 48 hours under normal circumstances. So these cells are completely replaced approximately every 2 days in your body. And that's hundreds of millions of cells, probably billions of cells actually. It would be absolutely billions of cells. The neutrophil number in our peripheral blood is about 1 million cells per milliliter. So it's a massive throughput of neutrophils, and also cells in the epithelium of our skin, of our gastrointestinal tract, of our lungs. And these cells are all turned over at a huge rate. So the massive amount of cell death going on in a lot of tissues, throughout our lifetime. And broadly speaking, there is basically the tissues that are exposed to the most damage or stress or infection exhibit the highest rates of cell death. So cells on the outside of the box, cells exposed to the world, essentially, to obviously pollutants and infectious agents and general wear and tear. These are the tissues in which we see cell death going on at a tremendous rate. And then more protected tissues in the interior of the body obviously still exhibit programmed cell death, but not quite as high rates as cells in the most exposed areas. But the point is, cell death and cell division obviously balance each other throughout life. We of course know that cell division is highly regulated. It's just that we didn't know that cell death was highly regulated until maybe 30 or 40 years ago. But of course, I hope I've persuaded you that there are lots and lots of situations that are very predictable where cells need to die throughout life. And so it's very important that cell death is also precisely regulated by a set of molecules that can be switched on in order to coordinate death of the cell when cells need to die. So there are lots and lots of situations during development. I'll give some examples. When you become an adult, even though tissues now are at a fixed size, there is a steady state turnover of cells going on for all the reasons I've mentioned in the adult. And then of course, in response to damage or infection, we see typically acceleration in the rate of cell death in the various tissues. So cell death control is very important throughout our lives. And here's a couple of classic examples. So the development of the digits, where initially digits start off as primitive paddles or buds. So in an embryo, the limbs initially develop from these primitive limb buds, which progressively enlarge. And then you have a paddle-like structure. And then you need to kill off all the interdigital cells in order to sculpt out the final form of the hand, the digits. And that type of sculpting takes place during development in lots and lots of organs. It's just very dramatically obvious, the formation of limbs. But the formation of blood vessels occurs in a similar manner. You make a solid a tube of cells, and then the center of the tube is cavitated out through programmed cell death to eliminate cells that you don't need. And in quite a similar manner, our brains, basically overproduce cells, neurons, and then you remove or eliminate the neurons that fail to make productive connections. And our immune system, famously developing T and B cells, are massively overproduced. And then they're educated in the thymus and in the bone marrow, in the case of B cells. And essentially, the new T cells that are produced migrate to the thymus. And then their T cell receptors are checked for fitness, to see that they work properly, that they're not autoreactive. And the vast majority of developing T cells, called thymocytes, are eliminated by apoptosis. So more than 95%, maybe 98% of the T cell population is found to be essentially not fit for purpose. So this kind of fitness test happens in lots and lots of tissues during development, where you get this overproduction of cells, and then you shape the tissue and select just the cells that you want. And again, cells are eliminated during that process by programmed cell death. This is not an accident. This is very much built into the system. And just to give another example here in the immune system, I mentioned earlier that when cells are infected, or maybe they're displaying peptides as a result of a mutation, peptides that are essentially that haven't been seen before in the body, well, there are actually professional killer cells called cytotoxic T cells, or natural killer cells, whose job it is to patrol the body, inspect the molecules that cells are presenting, either under MHC, either the conventional MHC molecules or non-conventional or atypical MHC molecules. And if they don't like what they see, then typically they have multiple mechanisms available to them in order to kill those cells, again, in a very targeted, very precise manner. So death takes place, throughout our lives. And as I've been emphasizing now again and again, it happens in a very regulated manner. And, sometimes this process fails. And when it does, we typically get disease. Now, this is where the death has been genetically intervened or manipulated. And here we have a mutation of a receptor that's involved in regulating the death of cells within the immune system. That receptor is called FAST or CD95. And when a mouse is created that has a knockout in FAST or CD95, what typically happens within a few months of birth, these are normal lymph nodes from a wild type mouse, and these are lymph nodes from a FAST knockout mouse that are about 50 times normal size as a result of the accumulation of all the extra T cells that normally would be turned over naturally through programmed cell death. Those cells hang around, and the same happens with the spleen. The spleen enlarges not quite as dramatically, but 10 times its normal size as a result of the accumulation of extra B cells. And then typically those extra T and B cells break tolerance. So they now start to recognize self-antigens, and these mice develop a condition like a human disease called systemic lupus erythematosus. So how do cells die? I hope I've persuaded you that cells need to die in lots of such situations. So cell death needs to be regulated because cell death is taking place, all over the place during development, and also when we're adults, helping to eliminate cells that we don't need, that we no longer need, that are damaged or aged or old in some way. So when that happens, when we need to get rid of cells, how do we get rid of them? And what are the consequences for not eliminating cells properly? So this is the way we used to think cell death took place. Cells, one day are going about their business, and everything's fine until something really terrible happens. And then that's it. The cell is dead and it's over. And that type of cell death, probably until the 1980s or 1990s, was called necrosis. We thought that all cell death took place by necrosis. That was the mainstream view, that cells die in a kind of a car crash manner. It's not regulated. They just kind of blow apart at some point as a result of being damaged. And in fact, that type of cell death does happen, but it's very undesirable when it happens. And you certainly don't want regulated cell death to take place like this. So I'll show you what necrosis looks like in a moment. But effectively what happens is cells just burst open as a result of, extreme damage to the cell. And that does happen in situations, for example, burn injuries, cells die by necrosis, where you have percussion injuries. In fact, if you are involved in a car crash, you're subjected to very extreme forces very quickly, compression forces. And, there's obviously a lot of necrosis as a result of that. And high concentrations of drugs or toxins also cause necrosis. And some infectious agents cause necrosis. So necrosis does happen. That's a very pathological form of cell death, and is not the type of cell death that we typically see in situations where death is kind of predictable, such as development, for example, elimination of T and B cells during, immune selection, or, for example, during wiring up of the brain, where you're eliminating the neurons that you haven't found productive connections with other neurons. And also during natural turnover of cell populations in tissue, we don't see this type of necrotic cell death. And we don't see this because necrosis is really very unfriendly. And as I'm going to tell you, it has a very detrimental downside. When cells die by necrosis, they rupture, and the immune system interprets this as a signal to respond, to get activated for very good reasons. And I'll tell you the molecules that are involved in doing that. So I'm going to be mentioning two main types of cell death: cell murder, which is necrosis, where essentially damage is inflicted upon the cell. The cell isn't the participant in its own death at all. It has no choice in the matter. The damage to the cell is so severe, typically cells die very quickly under those circumstances. And then cell suicide, or programmed cell death, is where a cell actually is an active participant. The cell is responding to signals or cues. It is sensing damage, that has occurred to the cell, or dysfunction, And the cell participates. It actually has molecules that, genes that are expressed and that are involved in executing the cell itself. So the cell is an active participant in programmed cell death. And normally, most programmed cell deaths are a type of a cellular suicide. Now, there's a kind of a subroutine to this where, in the context of infection or the acquisition of mutations, cells can be executed. That's also apoptosis. But in this case, there are other cells that are executing the cell, that are turning on the cell's own cell death machinery. So ultimately, cells are dying, of course. So cell death is the endpoint, and I'll tell you why it matters, why they die by apoptosis versus necrosis. So they are similar, and this is an old joke, but I'm going to show it because I didn't have time to delete it. So this person isn't dead. They're actually meditating, and apparently you put yourself in this kind of position. It's an advanced form of meditation. You stay there for maybe 30 minutes or an hour, and it's obviously quite good for stress levels and for your mind. And this person isn't meditating, but has ended up in a superficially similar position. And I think these are good analogies for apoptosis and necrosis. The endpoints are superficially similar, but the upstream stimuli are very different. And of course, the outcomes are very different of these two different processes. And I'm going to tell you the difference in terms of outcomes between apoptosis and necrosis now by telling you a little bit about what happens when cells die by necrosis. And remember, cells in multicellular organisms surrounded by neighbors. So, I told you about, it's very like human society. If your neighbor suddenly decides to kind of blow up their house, you're going to be affected by that. And you're not going to be very happy that if they just decide to do it themselves and they don't get experts in to do it, you're going to be directly affected. And in the same way, when a cell dies by necrosis, typically it just ruptures and it releases all of its contents. And of course, cells have a lot of nasty contents that are normally partitioned in organelles or kept inactive due to inhibitors that are complex with them. But when they're released into the extracellular space, now that decompartmentalization activates those molecules. So cells contain lipases that can obviously attack the plasma membranes of other cells, proteases, carbohydrases, DNases, RNases, DNA, not to mention molecules I'm going to mention now in a moment, which are called DAMs. They're effectively cytokine-like molecules that are normally never released from cells. But when they are released, they activate the immune system. And for good reason. They've evolved specifically to let the immune system know that there has been necrosis in the body. Because when you get necrosis, typically you're going to get an infection very soon afterwards. So the immune system needs to know when the body, when cells within the body undergoes necrosis, because typically, as I told you, necrosis happens in very extreme situations where there's severe departure from normal physiology. And typically, necrosis is either caused by infection or is very rapidly followed by infection. So there are molecules also released called DAMPs, damage-associated molecular patterns, and they activate the immune system. So you have all of this stuff coming out of necrotic cells. That's not good for neighbors. Typically, when you get a cell die by necrosis, then many cells die by necrosis around it. You get this big wave of necrotic cell death that radiates out from the initial site of damage. So that's not a good way to eliminate cells in a situation where just one cell in the tissue is not doing its job. That would be a very inefficient way of eliminating cells. So in general, the body tries to avoid necrosis because necrosis is very damaging and detrimental to neighbors. And as I've already mentioned, that the immune system responds very different to necrotic cells with respect to how it responds to apoptotic cells. So typically, necrotic cell death is responded to quite aggressively, and it's interpreted as being dangerous. And I'll just qualify why I'm using the word danger, because danger means something quite specific in immunology, and it was introduced by, or at least popularized by an immunologist called Polly Matzinger in the 90s, who wrote a very important review, really a hypothesis piece, which really, I think, changed how we look at how the immune system is activated. So Ubuntu, Matzinger's hypothesis was introduced in the mid-90s. It was essentially accepted that the immune system was activated as a result of the detection of infectious non-self. So what we mean by that is effectively non-self peptides or antigens were seen by the immune system, and the immune system, gets activated in response to them. But most immunologists know that that's actually not true. You can take a completely foreign non-self peptide and inject it into an animal, or indeed yourself, and you don't make an immune response. You don't make an immune response unless you have an additional qualifying signal that qualifies that peptide as being dangerous. And there are two main sources of that qualifying signal. One is some kind of conserved component of pathogens that many pathogens share. And we now know that those, conserved pathogen components are called PAMPs, or pathogen-associated molecular patterns. And these were known to be things that could quite aggressively activate the immune system, such as LPS. But it was also known that, cell death was also able to aggressively activate the immune system. So for some reason, you needed a non-self peptide along with these kind of, strong immune activators, or necrotic cell death. And Matzinger realized that the immune system really responds to is not so much non-self, but to danger. And the immune system has evolved to identify danger as being either necrotic cell death because of what they release, and I'll tell you what Matzinger proposed in a second, as well as conserved components of common pathogens that we now know, and they're called PAMPs. And we now know they're recognized by a suite of receptors that are basically pattern recognition receptors, such as toll-like receptors, nod-like receptors, and others. So the immune system responds to not so much non-self, but to things that are known to be dangerous to the body, and they're PAMPs. And then Matzinger introduced the concept that necrotic cells release DAMPs, or danger signals, she called them at the time, and they're damage-associated molecular patterns. She predicted that necrotic cells would contain molecules that are capable of powerfully activating the immune system if they were released into the extracellular space. And those things would tell the immune system that there's a lot of necrosis going on. And that's exactly when you want the immune system to be activated, when there's a lot of necrosis, because there's something really bad going on in the body. And so DAMPs are released from necrotic cells, and they're very powerful immune activators. And as I'll tell you in a second, DAMPs are probably, members of the interleukin-1 family. And these are molecules that typically are not released from viable cells, but they are released when you get necrosis. And they include molecules such as interleukin-1 alpha, interleukin beta, IL-18, IL-33, IL-36, alpha, beta, gamma, and also probably IL-37. So broadly speaking, what happens is when cells are healthy, the immune system ignores them. When you get necrosis because of the release of DAMPs, this is dangerous. This acts as a powerful immune activator. But apoptotic cells, as I'm going to tell you, they basically serve to shut down the inflammatory properties of a cell when it's dying, and effectively conveys a signal that says, look, I'm dying naturally, there's no need for an immune response. I'll just skip these slides in the interest of time. I already told you that what we think the canonical DAMPs are members of the extended IL-1 family. We've written a couple of reviews on this. One that's in press at the moment in cytokine, and one a couple of years ago in the FABS journal, and also in immunity. But there are other molecules, of course, as well that have been claimed to be DAMPs, and some of them are noted there. I'm a bit suspicious about those for a number of reasons that I don't have time to go into, but certainly we know that IL-1 family cytokines are probably major canonical DAMPs. So they're released during necrosis, and they're not released during apoptosis, and they're very good at driving inflammation. I'll skip this. Where we said this, so necrosis is inflammatory, apoptosis is non-inflammatory. Now, one of the reasons apoptotic cells are non-inflammatory is that when apoptotic cells die, they display molecules on their cell surface, and those molecules serve to allow macrophages to eat the apoptotic cells. So DAMPs are not released in apoptosis, because apoptotic cells don't ever get an opportunity to burst open. They're recognized at an early stage in the process as a result of the externalization of molecules such as phosphatidylserine on the surface of the apoptotic cells. And I'll touch on that later on. So just to summarize quickly before going forward, so necrosis is cell murder. Cell doesn't choose to die. Necrosis is typically caused by severe trauma, toxins, extremes of heat or cold, or radiation, nasty stuff. And this is what typically happens. Cells that are part of a tissue layer and epithelium, they detach, they swell very rapidly, and then the cell simply ruptures and bursts open. And it's very unfriendly to neighboring cells, as I've mentioned now. This is what it looks like. These are cells dying within 1 hour of exposure to 100 micromolar hydrogen peroxide. The cells quickly swell, and then you see these large blebs, and it just all burst very quickly. And the cell contents have been released. And not a lot seems to happen except just these plasma membrane blebs and ruptures. And then within an hour, all of the cells are dead. Apoptosis happens over a much more prolonged time period, typically over many, many hours. And I'll show you in a moment, it's very different. Oops. Okay, so apoptosis, as I say, it's very different. Cell chooses to die. So it's typically triggered by natural things made by the body, such as cytokines, such as TNF or FAS or TRAIL, or deprivation of growth factors or cytokines, but also by mild injury or damage that is sensed by the cell. So if there's a bit of damage to DNA, cells switch on transcription factor called p53 that synthesizes transcription or regulates genes that express proteins called NOX or PUMA that are able to switch on the cell death machinery. And of course, there are many other ways to damage cells. And so the cell usually is a participant. It actively controls the process. Now, the term comes from a group of pathologists in 1972 introduced the term when they realized that cells dying in some of the situations that I told you earlier had a characteristic appearance, and they recognized that this represented a kind of a turnover process, a healing process that was quite natural, just like when trees or leaves fall from trees in the autumn. And then during winter, of course, a tree looks a bit sad, but of course, it's still very much alive, and it's going to grow its leaves again the following spring. And it's just a natural part of the renewal cycle of the tree. And so the word apoptosis, they introduced meaning to drop or fall away, just like leaves fall from trees or petals fall from flowers. And of course, they wanted to choose a word that sounded like mitosis as a counterbalance to apoptosis. And that's where the word apoptosis comes from. Apoptotic cells exhibit a very characteristic morphology. I already touched on this earlier. These are apoptotic cells here. The cells are highly blebbed. They're smaller than normal cells. They condense, and the DNA becomes very fragmented. If you look here in the center, these are normal healthy cells stained with DAPI. The nucleus is very large and encompasses a large portion of the cell. In apoptotic cells, the DNA becomes very condensed and breaks up into highly characteristic fragments. Cells dying by necrosis, the DNA basically looks quite similar to a healthy cell. The nuclei do exhibit some changes, but there is not this condensation and fragmentation of nuclei as you get in apoptosis. And apoptotic cells exhibit many other characteristic features. They're highly blebbed, pieces shed off that enable phagocytes to eat the cell easily, and they're called apoptotic bodies. DNA becomes hydrolyzed into nucleosomal pieces, this characteristic ladder-like pattern of 200 base pairs, and many other features. And in apoptosis, typically cells detach, they condense in size, they undergo blebbing, the DNA becomes hypercondensed, it breaks up into lots of individual pieces, and then the cell breaks up into apoptotic bodies. And critically, during all this process, none of the cell contents are leaked out. And even from these early stages, cells exhibit membrane changes, such as the externalization of phosphatidylserine on the plasma membrane, that enables macrophages to recognize the cells and eliminate them. through phagocytosis before they can burst open and damage other cells, and before they can release stamps to activate the immune system. There's another view there. And this is what it looks like. This movie is shot over 24 hours. So the previous one I showed you of necrosis was over 1 hour. And here cells are individually making the decision to embark upon the process of apoptosis. And, you know, there are cells here that are, you know, exposed to the same concentration of drug for many, many, many hours longer than the others, and still don't die. They don't initiate the decision, they don't make the decision to initiate programmed cell death. So all of these cells are initiating, but they're still alive, they're not releasing contents. Now in vivo, if there were macrophages here, these cells would have already ruptured and burst open. But in vitro, in a tissue culture situation, of course, they still stay alive. They're manifesting apoptotic features, but they stay alive for many hours, and then eventually they will burst open if you leave them long enough. But in vivo, they'd be recognized as a result of changes that take place on their cell surface, as I told you earlier. So phagocytes and other tissue resident cells capture apoptotic cells and engulf them and eat them. And the reason is phosphatidylserine has really remained the only major molecule that's been shown from cells in all tissues to be characteristic of apoptosis. This molecule flips onto the external surface of plasma membrane early in the process, before the cell membrane you know, has become permeated in any way. There's other molecules that are released from apoptotic cells, such as the ATP and sphingosine-1 phosphate and certain chemokines that maybe guide phagocytes into the apoptotic cell to facilitate their removal. We can detect phosphatidylate externalization using a protein called an exon-5 if we conjugate it to FITC or another fluorescent dye. We can measure this early in the process. So this is a healthy cell population here. This is an exon-5 staining here on the x-axis, and this is propydium iodide, which measures membrane leakiness on the y-axis. So if a cell is in here, its membrane is leaky and it is binding an exon-5. So at the start of the culture, there's a small fraction of cells at the start of the experiment that are already, they've died, you know, over the previous like 24 hours. But now when you stimulate the cells to undergo programmed cell death, you can see that there's a lot of an exon-5 binding to the cell within a few hours, within 6 hours here of being stimulated, but these cells are not leaking. And then if you leave them long enough, of course, there's an increased fraction of cells that are leaking, but now almost the majority of the cell population are binding an exon-5, indicative of phosphatidylserine externalization. So that's one of the ways we can recognize apoptotic cells. So again, back to this point, because of the phosphatidylserine externalization, you get recognition of apoptotic cells by macrophages, and then macrophages eat the apoptotic cells. And typically we find when macrophages eat apoptotic cells, they release anti-inflammatory stimuli, whereas when they encounter necrotic cells, because of damp release, they release pro-inflammatory stimuli. So there's very different outcomes in terms of the response of the body to apoptotic cells versus necrotic cells. And then I've mentioned this cellular execution, where you have a cell and it might be paralyzed as a result of infected with a virus, and so it may not be able to turn on its own programmed cell death machinery. And in this case, natural killer cells or CTLs carry out, you know, the initiation of apoptosis. And just how this looks like now, typically they can either kill cells by deploying granules that fuse with the plasma membrane of the target cell, or by releasing ligands for receptors called FAST receptor or TNF receptor or TREL receptor on the surface of the target cell. They activate the programmed cell death machinery. This is again an example. This is a monolayer of transformed cells called HeLa cells, and the cells moving around are natural killer cells. This is an NK cell that's just come into the frame, and it's going to target this cell, and it's going to deliver a lethal hit by deploying its granules to kill this target cell here, and none of the neighbors will be affected by this. So so we'll die. Oh, actually, it's this cell that dies. Yeah. So it's killed this cell again, through this granule-mediated pathway. And again, we know very well the molecules involved here, which I'm going to go through in the next few slides. So I've shown you examples of programmed cell death and non-programmed cell death. So how does it all work? Well, some clues came from in the 1970s from working with the nematode Xenorhabdus elegans. This is a small worm, about 1 millimeter long. Of course, they live very naturally in the soil. They're everywhere. And Sidney Brenner in the 1970s, working at the Laboratory for Molecular Biology in the UK, was thinking about what kind of model organism would be useful to do genetics. You know, it needed to be something that didn't have too many cells, wasn't too complicated, and could be kept in the lab very readily. And he famously wrote a one-page letter to the Medical Research Council in the UK, asking for, you know, a major research grant to do work establishing the nematode. Xenorhabdus briggsei actually is what he originally started working on, not Xenorhabdus elegans, but then he changed to Xenorhabdus elegans later on. And that started off using the worm for many things, of course, not studying programmed cell death at all, because nobody knew about programmed cell death then. But during the course of work done by a postdoc in his lab, 2 postdocs actually, Bob Porwitz and John Sulzin, they were studying cell division and development in the worm. And sorry, I should have said that this is a C. elegans embryo at the very beginning of its life, and it's dividing here very rapidly. And then there's something very strange, that there were 1,000 cells that were produced, 1,090 cells produced, always during development, but always 131 of those cells died during development. And there's an example of a cell dying. Those disc-like cells that appear transiently, those cells died and then they were eaten by other cells. And they recorded this fact. They knew nothing about it, of course, but they, you know, they drew out these lineage maps where it showed cells, you know, they literally did hundreds of hours of microscopy where they would follow the development of Xenorhabdus elegans and trace the birth and death of every cell in the worm. And they found that there was always 131 cells that died during development. And they always died at the same time and the same place during development. And they published some early papers on lineage tracing in C. elegans. And then when Bob Horwitz went to start his own lab, he started doing genetic screens to see if there were genes involved in regulating this type of programmed cell death. And he found several different classes of mutants where genetically he could find worms that had too little cell death, so cell death was blocked, or there was too much cell death, you got excessive cell death during development, or as in this mutant here, the SED1 mutant, you had normal rates of cell death, but delayed burial. The dead cells were not removed the way they normally would be. And you're going to see this at the end here. On the right-hand side, you're going to see in the wild type, it looks normal. And then this mutant here, this SED1 mutant, that has these extra disc-like cells here because they haven't been, the dead cells have not been removed the way they normally would be as a result of this defect. So they had an engulfment defect. The macrophages failed to recognize that cells undergo programmed cell death. So over the course of 10 years or so, Bob Horwitz's lab established that there were about 11 genes that regulated this process, but 4 of them are really very central, and they call them cell death defective genes, SED1 through to SED10. And then this gene EGL1 was discovered in another screen. And they found that SED3 and SED4 were particularly important. They seemed to promote cell death, and SED9 blocked cell death. And EGL1 promoted cell death by inhibiting SED9. And that was their genetic framework. Now, of course, there was other work going on in mammalian systems that had also stumbled across the fact that, you know, cell death had to be programmed or regulated. So I mentioned earlier the work of Andrew Wiley and John Kerr and Alistair Curry, who introduced the term apoptosis, working on mammalian cells, working on humans actually. And there were, you know, people coming at this from different angles. And but certainly the first clue to the genetics came from work in the worm. And then other people took over really in terms of understanding, well, what are these gene products? What do they do? What proteins do they encode? And we now know that SED4 is essentially an activator. It's an activation platform. It's a molecule that can oligomerize into this octameric platform that recruits a protease called SED3. And SED3 is normally inactive in cells. And in order to become active, it needs to be clustered on the SED4 activation platform. But normally it's prevented from doing that because SED9 is binding to SED4 and preventing it forming this platform. So in a healthy cell, SED9 is sequestering SED4. And then when EGL1 appears, so in response to a stress or a stimulus that requires you to eliminate a cell, EGL1 is synthesized that binds to SED9 to neutralize it. And now SED9 lets go of SED4. SED4 now oligomerizes into this activation platform and allows SED3 to become activated. And SED3 is a protease. But precisely what SED3 does to kill cells is not really entirely understood. And we know more about the mammalian SED3 homologs in that regard. And as we're going to see, programmed cell death control is very similar in more complex multicellular organisms such as the fly, and of course ourselves, mammals. But there is a broad conservation of this basic paradigm. There's lots and lots of complexity in higher organisms, but broadly speaking, that's conserved. And Bob Horvitz and John Sulston and Sidney Brenner received the Nobel Prize together maybe 20 years later for their work, really on the use of the worm to dissect many biological processes. But strictly speaking, it was for the elucidation of the programmed cell death machine. But when they received the Nobel Prize, that was actually still many years before we really understood exactly what's going on. So just very quick summary now, looking at Drosophila melanogaster, the fruit fly, which is of course very often used as a genetic model. Things are more complicated. Now, instead of one SED3-like protein, you have multiple. There's actually 6 SED3-like homologs in the fly. Not all of them are involved in cell death control, of course. There's one major SED4-like homolog called DARK, which is a similar role. There are 2 SED9-like homologs. So remember, SED9 in the worm plays a very important role in preventing cell death normally. There are SED9 homologs in the fly, but they don't do anything in cell death. There's no role in the fly for any of the SED9 homologs. So instead, we have the appearance of these molecules called IAPs, or inhibitor of apoptosis proteins. And essentially, they set the brake on cell death. And they do so by binding to DARK, which is the worm, by binding to DRUNK, which is the fly equivalent of SED3. So again, we have, it's similar broadly, but there are a lot of notable differences that I've mentioned here. So in the fly, you have DARK, which is a SED4-like molecule. And normally, DIAP1 is binding to the protease, which is a SED3-like molecule in the fly, which is called DRUNK. And that keeps it inactive. So remember, we had SED9 binding to SED4 in the worm. Here we have the opposite. We have a molecule binding to the SED3 equivalent in the fly. And then when DIAP is eliminated, and it's eliminated by a number of proteins called repeated Grimmer sickle, the DIAP1 is displaced, and now you get a similar type of apoptosome formed where multiple molecules of DRUNK, which is a protease, are activated by the SED4-like molecule DARK. And then downstream, you get those proteases, cleaving lots of substrates in order to execute the cell. So again, it's similar but different. And in mammals, we're essentially basically a complicated worm. There's lots and lots of additional complexity there. But broadly speaking, you have a SED4-like molecule called APAF1. There's multiple caspases. APAF1 activates one of them, caspase 9. And then you have this SED9-like BCL2 family molecules, and then these EGL1 like BH3 only molecules, and they promote programmed cell death in that very similar structural framework. Now, it's considerably more complex. There's lots of SED9 pro-survival proteins, and EGL1 pro-apoptotic proteins. There's 12 human SET3-like molecules, which are called caspases, and one SET4-like molecule. Well, there's many SET4 homologs, but only one of them, APAP1, is involved in regulatory programs of death. There's also IAPs in mammals. And I don't have time to go into all the molecular complexity, but again, you have this apoptosome-like structure that's formed. And as a result of a lot of work, here's the three fundamental pathways we know exist in mammalian apoptosis. Three main ways of turning on the cell death machinery in humans and other mammals. You have death receptors. They're the extrinsic pathways, so they're molecules such as FAS or TNF receptor or trail receptor. When they're engaged with their ligand, they bind to adapters that bind to caspases such as caspase 8, which get activated. And then, surprisingly, you can then activate a BH3 only protein. So that's an EGL1-like protein that permeabilizes mitochondria. And in mammals, cytochrome C released from mitochondria is a critical checkpoint in cell death that activates the SED9 APAP1 or caspase 9 APAP1 apoptosome. And then you get multiple additional caspases activated. And then the caspases go on to coordinate the phenotypic changes that we see as apoptosis. Many other stresses or stimuli activate the so-called intrinsic pathway to cell death. So many stresses or damage to the cell activate a BH3 protein, and then they permeabilize the mitochondria by opening this back-to-back channel that releases cytochrome C that activates the apoptosome in mammals. And then CTL or NK cells, they can degranulate and introduce a granzyme, which is a protease called granzyme B that activates caspase 8 or BID to activate this pathway again. They also can kill cells by engaging the trailer FAS or TNF receptors. So everything now in mammals we know collapses down into three major routes to cell death: the extrinsic pathway, the intrinsic pathway, and the granzyme B or CTL-NK pathway. And I don't have time to go into this. I just have a few more slides now. I know I've spoken a lot quite far, so I'll take questions in a minute, but there's multiple caspases in mammals. I just want to give you a sense of kind of how things work. And they can divide it broadly into a pro-inflammatory subset and let's say an apoptosis subset, which, as I told you earlier, apoptosis is an anti-inflammatory mode of cell death. So they're really inflammatory molecules. They either promote inflammation or they shut it down. And they are activated during apoptosis because their active sites have these loops basically that are normally occluding the active site. And then during apoptosis, they get clipped to open the active site, and now the caspases can cleave their substrates. So how do they kill cells? Well, we don't really know. There's hundreds of substrates that have been identified. Here's a few examples of caspase substrates. I just want to give you a sense of what we think happens. So we know with some of the substrates, such as CAD, it's a nuclease that is normally inhibited by its inhibitor ICAD, and ICAD gets cleaved by caspases. That inactivates ICAD, and now CAD becomes activated as a result, and it hydrolyzes DNA to form the characteristic internucleosomal pattern you see during apoptosis. So there are hundreds of substrates. On the left here, we have healthy cell. On the right, these are apoptotic cells, where a number of years ago, we did these kind of global analyses of how many proteins are altered during apoptosis, and it's a lot, it's hundreds, if not over 1,000. And we've written a review in Nature Reviews a few years ago. And of course, we know the consequences of some of these cleavage events in terms of the impact they have on the phenotype that we associate with apoptosis. But I'd be lying to you if I told you we knew exactly how the apoptotic phenotype is produced. We just know that caspases are required to produce the apoptotic phenotype. And they kind of wreck the cell once they get activated during apoptosis. They cleave many, many different proteins that are responsible for the apoptotic phenotype. Now, mitochondria play this critical role in apoptosis in mammals by basically, they're the focal point for the assembly of this back-to-back channel that releases cytochrome C to activate the apoptosome. I won't go into the detail about this, but it's an important difference between apoptosis, how it works in mammals versus, invertebrates and how it works in insects and the nematode worm C. elegans that I told you earlier. There's no requirement for cytochrome C in lower multicellular organisms. So mitochondria play this kind of critical checkpoint role, and a lot of the program cell death machinery is kind of focused on mitochondria by opening this back-to-back channel. So cytochrome C is released in apoptosis in mammals and represents the point of no return in the process. And the reason why I mention this is that, in fact, so cytochrome C acts as a cofactor for the assembly of the apoptosome. And why I mention this is that, once you get cytochrome C release, cells are going to die whether or not you get caspase activation. And so caspases, strictly speaking, are not required for death in mammals, but they're required for the apoptotic phenotype, for controlling the kinetics of death and making sure that DAMPs are not released and coordinating the appearance of the membrane changes that, facilitate the uptake of apoptotic cells. And so this focus here, there's been intense focus on this interplay between the BH3 only or EGL1-like proteins and how they interact with and neutralize the anti-apoptotic or SED9-like proteins and how they open this back-to-back channel, because this is the real point of no return in the process. Once you get permeabilization of mitochondria and cytochrome C release, cells are going to die by necrosis if you block caspases, but by apoptosis if caspases are activated. I'll skip these slides now, but it's just to reiterate that caspase is required for apoptosis, but not death per se. You need caspases to control the phenotypic outcome of cell death. Skip that. Go back to this. So you have a healthy cell. Cell death triggers that promote programmed cell death. They activate caspases. And when you get caspase activation, caspases cleave lots of substrate proteins that basically ensure that cells die by apoptosis. And as I've already told you now, that guarantees that the cell death is largely immunologically silent or largely anti-inflammatory. By comparison with the alternative, if you block caspases or the death is not associated with caspase activation, you typically get this much more aggressive inflammatory necrotic cell death. And I will more or less leave it there. So final points are cell death and cell division need to be properly balanced throughout your life. If you get an imbalance, if you have excessive cell division relative to cell death, of course, we know that can lead to cancer and autoimmunity as you get expansion of cells that accumulate in compartments where they shouldn't. And conversely, if you have excessive cell death in a tissue that's accelerated, you can obviously have accelerated loss of important cell populations. And we know in many neurodegenerative diseases, cells die at accelerated rates in the brain, obviously leading to cognitive impairment. And just want to finish up by saying, even though of course we know a lot, and I started working in cell death during my PhD, so that's quite a few years ago now. And we've of course witnessed, just tremendous explosion of knowledge in this area over time. There's still a lot to discover. We still don't know everything, and we're looking particularly also the implications of cell death, for other cells and the immune system. That's a big focus in the last few years. And I put this up to illustrate that it's just like deep space, the inner space of cells is very complex. This is a view of a star cluster from the Hubble telescope. This is not a view from the Hubble telescope, but it's looking at protein clusters just in one tiny fraction or compartment of one cell. As we zoom out, all of these different clusters are protein machines doing things that we clearly don't understand yet. And this is just visualizing a few of the proteins in the cell. So I think we're still at the stage of, a pretty low resolution understanding of how cells work and how they die, how they live, how they divide. Still lots of questions we can ask. And I'll leave it at that. And just a little plug for, if you're interested in immunology, a great textbook called Essential Immunology, which I've been involved in for a while. Okay, thank you.
Many thanks, Seamus.
Thank you,
Again,
Seamus.
wonderful introductory lecture. I will ask the students to turn on the video and start to ask questions, as well as people from the YouTube. The YouTube people can write in the chat. So any of you students want to start?
I
Or
realize I went through.
I can start.
Yeah.
no, Karina, wait, I think somebody is. So you can either raise your hand, I think, with this reaction button, or you can just open up the microphone and
Yes.
ask a question. I think it was.
Abolade.
Oh, all right.
Yes.
Go ahead, Bolaji.
Okay. So thank you very much for this presentation. I've really learned a lot, but however, I would just like to ask a question that is not a bit clear, just the activity of SED and the EGL in apoptosis. I don't know if you can just give a clearer explanation on that.
Of the activity of SED3.
Yes, please.
SED3 is a protease, so proteases cleave other proteins. So normally SED3 is inactive in the cell, like most proteases. And then when programmed cell death is initiated, SED3 gets activated by binding to SED4 because it gets clustered with other SED3 molecules and they're able to activate one another. So once SED3 gets activated, then it can cleave lots of different proteins in the cell and change their function. So that's how SED3 activation works.
Shay, I think I will jump in some of these questions just to, maybe increase a bit the scope of the questions. All right. So you did a lot of work on the substrates, either for the caspase 3, which is the central. You did also caspase 6, 7. You did excellent work on caspase 1 in the mammalian, and of course they are different not only in the specificity but also in the complexity. What about SED3? I don't remember. I think it was you did also in your lab with
yeah, we did a screen for SED3 substrates, and in fact we found quite a number of SED3 substrates in worm cell lysates. But as I've said, so there are dozens of SED3 substrates we found, but it's complicated. As I've said earlier, we don't really understand in any kind of detail exactly the consequences of the cleavage of individual substrates and what that leads to in terms of the cell death phenotype, because there's hundreds of cleavage events going on all at the same time, and it's very hard to disentangle exactly what each individual cleavage event represents. Now we know what some of them do, of course. Some of them cause quite specific events, but it's a complicated process. It's a bit like this. The way I look at it is a bit like this. Imagine you were you were kind of moving house, right? You were demolishing your house. You were going to move to a completely new house. You're going to take out every item in your house that you need to live. The table, the couch, the TV, the kitchen, the cooker, the pots and pans, everything. And of course, you need all of those things. And you're going to take them all out and move them to a new house or apartment. It kind of doesn't matter the order in which you take them out. And none of those specific things are critical to live individually, but collectively they're all important. And apoptosis is like taking out all of the kind of vital, let's say, processes and pathways in the cell, none of which though are absolutely on their own completely vital. but all of them are important to kind of extinguish. And many of the events ultimately, I think, go towards just shutting the cell down in a kind of a progressive, controlled manner, and ensuring that, you know, the changes that take place on the cell surface that facilitate the removal of the cell take place. So it's a complicated process. And in biology, we often look for, in research, we often looking for, you know, things that cause very dramatic binary changes in the cell. You know, if this doesn't happen, then this other thing stops. You know, if you don't get proteolysis of this substrate, you don't get this outcome. Well, it's not like this in the majority of cases. And that's why at a certain point, unfortunately, we stopped looking for caspase substrates and trying to understand the process, because after having done dozens and having mutated those cleavage sites and then reintroduced non-cleavable mutants and finding a very modest impact on either cell death outcome or phenotype, at some point you get into diminishing returns on your investment. And you then say, well, look, somebody else can do that work. So that's why.
Right.
But as I said, we summarized, you know, we're 10 years ago now. Here's what we know in terms of, you know, what caspase substrate cleavage event leads to what outcome. And we put together, I think, a pretty decent overview. It's certainly been fairly highly cited a couple of thousand times now on how caspases kill cells. But we don't know in fine detail exactly how they do, or how at least they contribute to the shutting down of the anti-inflammatory or the inflammatory properties of cells. But we know some of the things.
Yeah, but that's exactly what students keep asking us, you know, in the many, you know, courses that we mention or we give on cell death, that, you know, what would be the crucial substrates? And I think your answer clearly, you know, tell us that there's no one specific episode.
Yeah, it's like asking, what's the most important thing in your house? You know, what is it?
Yeah.
What is it?
I like very much, you didn't mention here, you mentioned a different analogy about removing, you know, things from the house, but I very much like your first review article on caspase, when you talk about this tent, you know, the circus tent, and then if you cut just a couple of you know, wires that support it. Can you just mention that before, because I think it's very
I mean, you could say caspases are kind of cutting the structural supports that keep the cell up, keep the cell alive, and like a kind of a large tent, which is held up by you know, several poles and many ropes. If you cut one or two ropes, the tent still stays up. You know, you maybe destabilize it a little, but the more ropes and poles you cut away, eventually you get a complete collapse of the tent. But no individual rope or pole is absolutely critical to keeping the tent up. So caspase substrates, are quite like that. And, you know, some of them play maybe slightly more important roles than others. And they might be the major support poles in the tent, as opposed to the ropes.
Cool, thank you. Samson.
Okay, thank you very much, Professor Martin, for the wonderful lecture. Okay, I quite have two questions here. The first one is, you mentioned initially that apoptosis is like a kind of voluntary action from the cell, from the dying cells itself. But later you mentioned something like involuntary apoptosis, so I wasn't quite getting it right. So I don't know if you could explain about involuntary apoptosis.
Yeah, so most apoptosis is controlled by within the cell, but then I mentioned a specific category of apoptosis that is caused by cytotoxic T cells or natural killer cells, where a cell is infected with a virus or maybe a bacterium, and the virus or bacterium is preventing the cell from turning on its own cell death machinery. And in this case, cytotoxic T cells or natural killer cells, they execute the target cell. And that's also apoptosis, but they attack the target cell and switch on its cell death machinery. So that's a special example of, let's say, apoptosis that's involuntary, but most apoptosis in the body would be voluntary. The cell senses that it's injured or damaged or aged in some way, and it initiates its own programmed cell death machinery. So yeah, that's what I mean by that. So cytotoxic T cell or natural killer cell apoptosis would be the involuntary apoptosis I was mentioning.
Okay, thank you very much. The second question, you were comparing apoptosis and necrosis in the presentation. So you talked about when macrophages eat up cells that are necrotic and after then releasing inflammatory signals. So when you talk about apoptotic cells, when macrophages eat up apoptotic cells, they release anti-inflammatory signals. So I was wondering, to what end is these signals released? I mean the anti-inflammatory signals, because for the inflammatory signals, we know, okay, they will recruit more immune cells to actually finish up the work, the cell death process. But in case of apoptosis, when anti-inflammatory signals are released, to what end is this? So that's the question.
To what end?
Yeah.
Well, the end in that case will be to ensure that you don't get activation of inflammation in the tissue, because when you have a lot of apoptosis going on, some cells could end up rupturing a little and releasing some DAMPs. I mentioned that if you, for example, if cells die by apoptosis in vitro with no macrophages around, they do eventually burst open. So they will eventually burst open if you leave them long enough, because of course caspases are active, shutting down all of the kind of cellular systems. And eventually you will get what's called secondary necrosis if you leave the cells long enough. So in a tissue where there's a lot of apoptosis going on, it could be important for when macrophages eat apoptotic cells to secrete anti-inflammatory signals to ensure that if there is any secondary necrosis happening in the tissue, that you don't get a full-scale inflammatory response. But you know, that's speculation. And actually, in terms of the anti-inflammatory response to apoptotic cells, I'm not sure there's really a lot of work that's been done in that area. There is work that's been done in the area, but I think in a lot of cases, when macrophages eat apoptotic cells, they are just quiescent. They just don't release any inflammatory signals, or or they release an anti-inflammatory profile. But at least conceptually, I think if you get this anti-inflammatory response, it may be to quell any response to secondary necrosis that might be happening in the tissue at the same time. So, yeah, that's mine.
All right, thank you, Samson. Thank you, Seamus. Just before I leave
Move on to Lucas.
Yeah, there's a question in the YouTube, Seamus, from
Yes.
our friend Marco Ataidi. So I'd like to also thank everybody that is joining us through the YouTube. Seamus, Marco was saying, regarding the dead cell clearance phenomenon, could you expand the discussion in the light of the role of NINJ1 in the plasma membrane rupture? When would NINJ1 be required for the dead cell clearance? Would NINJ1, for example, be needed in the thymus during T cell selection? If so, why wait until the membrane rupture?
In terms of necrosis?
Yeah, I think in terms
of cell death in general, in terms of, how can I say, clearance phenomenon.
Yeah, really, to be honest, in terms of NINJ1, so I'm not sure there's any data suggesting that NINJ1 is involved in apoptosis at all. In terms of necrosis, which is, I think, the only paper in NINJ1 that I'm aware of from Vishwa Dixit's lab, it is concerning necrosis. So yeah, in terms of its role in that particular context, I don't think we know enough yet about NINJ1 and the type of cell death that it participates in to really know in any kind of informed detail about, you know, how important it is. You know, is it involved in all forms of necrosis or not? You know, this could be just a very special subset of necrotic cell deaths. So I can't really comment in any detail on that, you know, but I very much doubt that NINJ1 is involved in thymic selection or in, you know, in any context where it's the cell death is apoptotic in nature anyway. So I think it's just too early to say what role NINJ1 plays, you know.
Yeah, but if I can also, Marco and Seamus joining in the discussion, I think Vishwa also clearly show that NINJ1 is very important for the cell to burst open during pyroptosis and release this large molecular weight proteins, but not even for the release of small cytokines like the IL-1 family members that you mentioned. And this is not true for necroptosis. I think NINJ1 is not important for the burst and the release of large molecular weight proteins for necroptosis. So I think it's particularly, sorry about this airplane here, but I think is particularly important for pyroptosis. I don't see as well.
No, NINJ1 is important to cell rupture, not to cell release of the molecular, different molecular weight. It's only...
no, I mean, he mentioned, Karina, he studied the role of the release of like LDH compared to IL-1 and so on. So apparently gasdermin pores are enough to release IL-1 family members, but not LDH, for instance, during pyroptosis. Yeah. So then the cells need NINJ1 to release LDH during pyroptosis. But when he Vishwa assessed necroptosis, there's no need for NINJ1 to get the release of LDH during necroptosis. So what I think I was trying to say is that I think to Marco is that, and then to, of course, to the other students, is that NINJ1 seems to be important during pyroptosis. I agree with Seamus. I don't see any role of NINJ1 during any apoptosis event, including the one that Marcus specifically mentioned was the thymus development. That's what I was trying to say, in my view, at least.
I think we shouldn't overstate the importance of a single protein, NINJ1, that just happens to be the newest kind of protein. And you know, in the NINJ1 knockouts, the cells still die. It's just that kinetically, the release of certain molecules is a bit slowed. So it's kind of cute. It's a molecule that's clearly susceptible to permeabilizing when you get, you know, ionic fluxes, you get damage to the membrane, and it facilitates the rupture. But let's not overstate this here. NINJ1 could turn out to be an unimportant downstream player, and you know, it's interesting, but just because a paper is published in Nature doesn't mean it's, you know, that important in the fullness of time. So it could just turn out to be that NINJ1 is a protein that has a propensity to oligomerize upon kind of the ionic fluxes that are typical of things that cause necrosis, and so it speeds the process up a bit, you know. Like having the sunroof on a car during a car crash. You know, it rips the roof off the car a little bit quicker than it would otherwise. But, you know, it's not the be all and end all. You still got the car crash taking place. So.
All right, just closing this question, Marco said that in the paper they say apoptosis, necrosis, and pyroptosis, but Vishva, I saw a couple of lectures from Vishva after the paper was published, and I think
They don't say
he
apoptosis in the paper.
yeah, he's really means in the pyroptosis. Anyway, let's move to Lucas Vasconcelos has also a question. Lucas.
Hello guys, hi Seamus. Thank you so much for that class. It cleared out a lot of things for me. I have two simple questions. The first is, there is one or many proteins that can make the phosphatidylserine to flip from the inside of the membrane to the outside. I didn't get that right from the lecture.
You mean that? Well, there's probably at least a few, but Pennexin 1, I think Pennexin 1 channels have been implicated in this, and I'm not sure whether that's the whole story. There's been not a huge amount of work done in that area, but Pennexin 1 has been implicated. It's the proteolysis of that. I'm not entirely convinced by that data, I have to say, but Cody Ravichandran has published a number of papers in this area, and there may have been other papers in more recent years that I've missed, but as far as I understand, Pennexin 1 proteolysis has been implicated in causing the destabilization of ionic fluxes in the membrane that lead to phosphatidylserine redistribution. But I don't think there's a satisfactory molecular explanation exactly for that right now. But we do know it's caspase dependent, because we know that we can initiate apoptosis, and if we block caspase activity using just globally specific, pan-caspase inhibitors, then we can block the phosphatidylserine externalization for many hours. So we know caspases are cleaving something, but we don't know the detailed molecular mechanism of that. But Pennexin 1 has been implicated.
I got it. Thank you so much. And my next question is like really simple, but I just got curious. In an in vitro environment, how long could it take for the cell to respond and to finish apoptosis without any external stimuli?
Without any external stimuli? Well, why is it dying? You're just saying if a cell was
like if you give it a drug like a
drug.
Yeah, if you give it some drug for the cell, yeah, like deacetylase inhibitor or a BTK inhibitor.
Yeah, it really depends on what you're hitting in the cell. What kind of drug it is. The dose, what particular inhibitory effect it has. But for example, if we give cells very high concentration of, for example, TRAIL, which is a ligand for the TRAIL receptor, which is a plasma membrane receptor, we know exactly how it kills by apoptosis. We can start to see cells dying if we put very high concentrations in within 3 or 4 hours. Now, if we put more physiologically relevant concentrations, it will take more like 18 hours. So it really depends. It's titratable. If you use a high dose of a pro-apoptotic stimulus, then usually you can get cell death within 6 or 8 hours. If you use a more moderate dose, the cells will take 24 hours to die. There's certain stimuli that we've used that the cells look reasonably. So if you block the proteasome with low dose of proteasome inhibitors, and at 24 hours, there's maybe only 20%, 15% of the cells are dead at 24 hours. But at 48 hours, they're all dead. So depends on how you damage the cell or what kind of stimulus you give to the cell. But typically speaking, if you give a high concentration of injury or stress, cells will die rapidly. And if it's a low concentration and more chronic, they'll die slowly. So.
Seamus, can I jump in again? Because also we've been receiving this kind of questions. So how long it takes for a cell to die by apoptosis, and you clearly show in your videos that when you do like a necrosis, it's so dramatic that everything is more or less synchronized and occurs faster. But yeah, even if you like drop like a staurosporin, actinomycin, one of these apoptogenic stimuli in a cell line that's supposed to be genetically identical, they, as you just mentioned, they do not undergo apoptosis in a synchronized way. So why exactly is that, or how many factors contributed from the the amount of drug that the certain cells receive it, and other like difference like epigenetic or fitness of individual cells in the same culture? Can you just discuss a little bit on that, please?
Yeah, well, it's like ourselves. Like if you have a closely related human population, no individual will respond exactly the same to an external stimulus, and so it's a mistake to think that cells are genetically identical in a tissue culture setting. They're not. So for starters, cell cycle position can have a bearing on the outcome. Cells at different stages of the cell cycle have a greater or lesser vulnerability to getting damaged. So if you get a cell going through mitosis, it's very vulnerable when it's going through mitosis. And so if you damage it at that moment, death will very rapidly follow in mitosis. I mean, we know that's been shown. And similarly, if a cell in S phase is very vulnerable, if it's synthesizing, replicating its genome, in S phase, you can't go back. When you initiate S phase, you can't half replicate your genome and then decide, oh, I'm getting damaged. Now I'll just stay here, or I'll go back now. I'll go back to, just 2 N chromosomes. So cells in S phase are uniquely vulnerable. And of course, the cells as they go through the cell cycle express different cohorts of protein. So that's one factor. Another factor is, of course, there is a stochastic variation in any cell population of, the exact number of pro-survival BCL2 family proteins a given cell will have, due to, epigenetic differences, due to also the experience the cell has had in, even though broadly speaking, it's, of course, you would think it's the same environment. It's not exactly the same environment for all cells. So, the totality of its pro versus anti-apoptotic BCL2 family proteins, is going to have a bearing on the outcome. And also the exact quantity of drug that the cell will receive relative to another, so maybe if you treat cells with a drug, one cell will get 1 million copies of the drug, and the other will get 900,000 copies. And so, that's going to cause a slight difference in the outcome, especially if that drug is sensed, that you turn on transcription factors that then go and turn on other genes that dictate the outcome. Cells have cell stress sensing pathways where the response to stress is to try and mitigate the stress, by turning on the specific stress sensing, either the DNA damage response, the unfolded protein response, the heat shock response, the reactive, oxygen response, and so on. And all of these are transcriptional pathways that, they're again, themselves are highly dynamic, highly regulated. They're scaled, they're not all the same. Cells don't, just like ourselves, we all don't act as, a clone, in a colony. Cells are not clones of one another. They have different differential expression of genes, of proteins, and again, they, if you add a drug or a compound or a cytokine molecule, one cell may get, as I said, 100,000 TNF engaging the receptors, and another might get 80,000, and that threshold difference can have fundamentally different outcomes. So just in the same way, I'm going to illustrate this the way I do sometimes. I'm now dropping the threshold. See?
All right.
So I just dropped the threshold of my voice,
You're right. Yeah.
to go below a threshold that was actually biologically significant. So, you know, in the same way, all biology is about thresholds.
Excellent, excellent.
Let's
There's
move to David, and after that, Lais Macchioro did a question here.
I think there's a question. I didn't.
David is.
Oh, David. Okay.
Yes.
And then we have Amanda and Lais in the chat. Okay. Go ahead, David.
Can you hear me?
Yep.
Thank you so much, Professor Martin, for your presentation. Recently, I started to work with the Omicron variant, and we noticed that the cytopathic effect caused by Omicron, it's way different from the other variants. Like it takes way more time to see the cytopathic effect. So I got curious, how can we know that when the lower cell death in infected cells, it's a good or bad thing for the host?
How can you know? Well, I suppose you know by experiment. You can't know, you know, by default. You know, we have this debate with many viruses. So it can be a good thing, you know, in many respects. It's a good thing if a virus infects a cell, it's detected and the cell dies very quickly. It shuts down the viral infection. The virus doesn't have time to replicate and you just contain it. But the problem is if you have that happening in a very critical cell population, then that can cause a lot of problems. And it's, you know, so you may basically prefer for not much cell death to happen, and then maybe later on you get cell death, you know, happening at a bit of a slower rate, you know, to eliminate cells. But that also can be bad depending on the scenario. So, for example, HIV can be latent for years in an individual because the cells don't die when they're infected with certain strains, and they may get reactivated at some point in the future, and now, you know, you have a persistent viral infection that won't go away. So I don't think you can generalize about whether it's a good thing or a bad thing, whether the virus kills cells quickly or not. It's case by case. It depends on the cell population of virus infecting. It depends on whether that cell death is caused directly by the cytopathic effect of the virus, or is it a response to the virus by the immune system or by the cell that's been infected. I think you have to see what the consequences of that are. I think conceptually, I want my cells to die immediately when they're infected with a virus, so that the virus is immediately eradicated from the body and doesn't get an opportunity to replicate and establish a clinically significant infection. But clearly with SARS-CoV-2, that's not the case. The virus gets a foothold, it replicates, it produces virions quickly. You know, with Omicron, so you said it kills cells less rapidly than other variants. Is that the case? Well, that could, you know, that's probably a good thing in this particular case. You know, it gives the immune system time to clear the virus, and, you know, we don't get as ill, obviously. But you know, I think it depends on the situation, really. Remember, this is a virus that's adapting, that's evolving to adapt to a new host. So it's complicated. But I think viruses generally evolve to cause as little cytopathic effect in the host as possible, because they're not very successful otherwise if they don't adapt to the host. At least thinking about it again anthropomorphically, any successful parasite should become highly adapted to its host and not cause the host to make a big response. And I mean that by, look at the response that this virus has elicited from society, a massive response, right? That's a very badly adapted virus. So ultimately, when it's very highly adapted, I think it will cause minor cytopathic effects, and we will tolerate this virus just like the common cold.
Okay, Shay. I will do the Lais question here in the chat. So in terms of aging, is the contribution of telomeres, for example, the same pathway on apoptosis, the same activation pathway?
I'm not really sure what the question is. The question, are telomere shortening linked to apoptosis? Or is it that, you know, it's kind of, are there parallels between the two processes? I'm not really sure. What's the specific question in terms of
aging?
Yeah. Well, I don't think we know a lot about how telomeres or how telomere shortening results in apoptosis, if that's what the question is, in the context of aging. So the question might be, as telomeres shorten, of course, which happens cells age, that normally leads to replicative senescence, where the cell effectively now is no longer able to divide. And is somehow telomere shortening, then does it lead to apoptosis? I think generally not, because cells typically undergo senescence as a result of telomere shortening, not apoptosis. So that would be my understanding. Now, ultimately, senescent cells do die, and they probably die by apoptosis. And that's not a lot of links. There's not a lot known about that right now, the links between senescence and apoptosis, although there is work going on in that area at the moment. It is known that one of the factors that can that's important for that switch to senescence is you know, BCL2 family proteins. And we know that because one way of getting rid of senescent cells, at least, is venetoclax has now been promoted. That's a BH3 only mimetic. It's a BCL2 family protein inhibitor. And venetoclax is now being kind of suggested as being a new class of compounds that are called senolytics. These are drugs that are designed to get rid of senescent cells, which are considered to be problematic, particularly in the context of cancer, but also just generally problematic in tissues and contributing to long-term chronic inflammation. So there's kind of a lot of interest in that area of senolytics. And there seems to be some links between senescence and apoptosis, but what the links are explicitly are still not really understood very well. But in terms of links between telomere shortening, I think typically results in senescence rather than apoptosis.
Okay, Shay, thanks. Amanda asked, great class. Do you believe that this process of cell death, specifically neutrophilic apoptosis, can be delayed, leading to even more severe disorders? Do you believe that that is possible to have therapeutic targets inhibiting this delay?
Do I think it can be delayed in disease conditions? Yeah, I think there's certain conditions where physiological cell death is delayed. We know that neutrophil cell death is prolonged by pro-inflammatory cytokines, for example. And inflammatory neutrophils typically live longer. And I'm sure there's lots of other situations where you get a delay of cell death. We know that tumors effectively, as a result of overexpressing BCL2 family proteins, they delay their death. They're not immortal, but certainly they live a lot longer than they should. And you know, there of course are other situations. So T and B cells that should have been deselected during time of development or just in the periphery, it seemed to their death seems to be perturbed under certain circumstances. They don't become eliminated the way they should, triggering autoimmunity.
And neutrophils can also delay the apoptosis and became in a hyperactivated state. It's well known also. I think neutrophils has a membrane repair more, I don't know, quickly than macrophages, for example, in some situations.
Yeah. But in terms of therapeutic targeting, I think that was part of the question. I think whoever asked that question was asking that as well.
Yes.
So yeah, I mean, I mentioned already venetoclax, BH3 only proteins are a group of compounds that, sorry, I'm getting a phone call here. A group of compounds that that neutralize the pro-survival proteins and can accelerate cell death. And venetoclax, probably the first apoptosis targeted, therapeutic compound that was introduced, I think, into the clinic about 2016, treated a number of cancer types. And there's a whole range of other targeted therapies that are targeting the BCL2 family proteins that are basically in clinical trials at the moment. So there are ways of accelerating cell death or overcoming the delay to cell death that certain populations manifest.
Yes.
And of course, there are other molecules that, we didn't realize maybe the problem they're causing is cell death, such as TNF, for example. TNF is overproduced in many autoimmune disorders, and TNF has two outcomes. One is to promote production of inflammatory cytokines, but the other is to promote cell death. And it's not, we always thought that TNF was problematic in terms of promoting inflammatory cytokines, but maybe that's not the case. There's increasing evidence that TNF's cell death inducing function may also be important in immune disorders. And of course, TNF neutralizing antibodies and receptor antagonists have been in the clinic for more than 10 years and are extremely successful at treating rheumatoid arthritis, Crohn's disease, IBD, psoriasis, and many other autoimmune disease. And that's effectively, you could call that an anti, cell death effector molecule strategy. So.
Okay, Shay, let's go to Gustavo Henrique. So,
I think you are mute.
You are muted. No?
Everybody, can you hear me?
Okay.
No?
Yes, we can.
Yes.
Professor Martin, thanks for the lecture. My question is regarding clinical research. I am currently working with biomarkers of apoptosis in humans, and I've seen some works that assess apoptosis components like cytochrome C on plasma or serum blood, on blood of humans. My question is this biomarkers very inspecific for chronic diseases? For example, I don't know if these biomarkers reflect the damage caused by chronic diseases, or if it's related with immune response. I don't know if it's very...
Yeah, it's hard to know. I mean, that, for example, using cytochrome C, it's not a biomarker for apoptosis because it's course released from necrotic cells as well, and any cell that dies, that ruptures, will release cytochrome C. So it depends. Look, I don't know any really very useful. Cytokeratin 18, I think, is used in some studies as a kind of a circulating biomarker of apoptotic cells, because I think a particular cytokeratin 18 cleavage product that's cleaved by caspases can be selectively recognized. So that might be a reasonable apoptotic biomarker, but it's not a straightforward question to answer. So, I can't give a clear answer to that. I don't know whether the biomarkers, you're using in your particular study would be a good reflection of chronic disease or not. I'd have to see what they are and have a think about it, to be honest. So, but I often see studies claiming biomarkers of this and that, and then when I read the data, I laugh. So
Thanks.
okay, it's a complicated thing though to measure.
Yes.
All right, people, we are on the edge of our timing. So I'd like to thank Seamus again very, very much for this brilliant presentation and all his input and suggestion and discussion. We will break. I want to also welcome Tommy. Tommy is already on the room, but we will break for 10 minutes before we join Tommy and Karina formally introduce him. Also, I'd like to tell everybody at the YouTube that we will stop our transmission, and the next lecture will be only held in the Zoom room, okay? And so for those in the YouTube, we will get back tomorrow morning. All right, Seamus, thank you so much.
Thank you, Shay.
Okay, everybody.
Marcio, if you want to break